From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on April 10th, 2025. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 87th episode of Matters Microbial. I am truly grateful for every download, listen, and view on YouTube. Thank you all for spreading the good microbial word far and wide. First, I am quite excited about being invited to the University of Texas, Austin later this month to give a seminar, put on a LuxArt session, and hopefully have a remote Matters Microbial podcast. I'm very grateful. My wife, Dr. Jennifer Quinn, and I created these glow-in-the-dark buttons for LuxArt participants, and I'm, of course, bringing plenty of microbial swag to hand out. As this session is being recorded, students here at Puget Sound are registering for my fall microbiology course, probably the last time I will be teaching microbiology. So I'm thoughtful about what to have them do in lab to really make an impact. Plants have a complex metabolism and actually emit small amounts of methanol through their stomata. Naturally, some microbes can use this form of alcohol as a carbon source. So when the leaves are printed onto selective media, the image of the leaf appears due to pink pigmented facultative methylotrophs. It's a wonderful reminder of the microbial world in which we live. I will put a link to these organisms in the show notes. During several prior episodes of this podcast, we've had guests discuss the microbiome. Because of the importance of the microbiome, it would be interesting to use modern tools to determine not just the effects the microbiome has on the organism, but what effects the organism has on the microbiome. Kind of a play on you are what you eat, microbiome-wise. Now, this includes the impact of diet. So I'm grateful to welcome Dr. Manuel Kleiner of the Department of Plant and Microbial Biology at North Carolina State University to the podcast to discuss the fascinating and diverse interests of his research group. Welcome to the Quality Quorum today, Manuel. It's great to meet you.
Thank you. Thank you so much for having me, Mark.
The first thing that I want to tell everybody is that Manuel's research group has about a gazillion types of interests, and there will be a link to that, but we're going to focus on just one thing for today. And it's hard for me because I want to talk about everything. So I just want to applaud what you and your group do.
Yeah, thank you. That's true. I do like to do a lot of different things. Just now when you talked about methanol, I was like, yeah, we also do something with methanol in plants, right? It's not what we're going to talk about today, though.
Yeah, specialization is for insects, is the saying. But, on the other hand, sometimes you do need to drill down. And actually, I read some of your papers, and you do indeed drill down on many things. So the topic you're discussing with us today, can you get us started?
Yeah. So today we'll be actually talking about the interactions of different dietary components with the microbiome in the gut, and ultimately what that might mean for host health. And the topic that we are most interested in is actually dietary protein. So we're really looking at dietary protein, different sources of dietary protein, and how they interact with the microbes in the gut. So for this work, we work both with mice. So we have mice where we can, have a lot of control over how we work with these mice. Mice without a microbiota, mice with a microbiota. So there's a lot we can do, but we also actually do some work with controlled feeding studies in humans.
So for listeners and viewers, can you tell folks why it is, on the one hand, working with mice is valuable versus looking at humans?
Well, ultimately, of course, we're interested in health impacts in humans. Now, of course, there's a few things that we cannot easily investigate with humans. And so that's where the mice come in, right? It's much easier to give a very controlled diet where we define every component to mice over a longer period of time. I think it would be very hard to recruit humans to, say, for 10 days to eat just one protein, one fiber, and maybe sugar, That would be very difficult, right? So with mice, we have that ability to feed them a very controlled diet over time. And then, of course, we can work with mice that are genetically inbred. So they're all genetically very similar. With humans, we always have a huge amount of variation. So if you do work with humans, you need much larger groups of humans to actually get rid of the individual variation. Whereas with mice that are genetically bred to be very similar, you have at least that one factor that you can remove from the equation and really focus on your topic. And then, of course, sampling is ultimately sometimes easier with mice, right? If we need, in our case, we work a lot with stool samples. They can be obtained from humans, but it's much easier to obtain them from mice.
You also mentioned parenthetically the idea of organisms without a microbiome. So what you're referring to are notobiotic or, quote, I hate this term, germ-free mice?
Yes, so I'm referring to germ-free mice, so mice that have no microbiota. They're actually a specialized version of notobiotic mice. So notobiotic is actually the term that we use when we say we have an animal or a plant where we know all its constituents, right? So we know all the microbes that are in there. And of course, the simplest case of that is an animal without any microbes. So a germ-free mouse is essentially a notobiotic animal, but the simplest form of it.
Well, I want to thank you for that idea, because I apparently have not been using that term correctly for many years. So thank you. I presume that what you're doing is taking, looking at, as we've talked about many times on the podcast, one way or the other, taking, for example, a stool sample, isolating total DNA from it, and doing deep sequencing, looking at perhaps 16S regions?
Not quite. So that's actually where we really shine, because we use a method that allows us to dig much deeper than that. Right? With what you just mentioned, 16S sequencing, we have the ability to say who is there, and maybe we get also a sense of how many different microbes and how abundant they are. But it really doesn't tell us much about what they are doing, which is kind of the question we are really interested in. What are the microbes in the gut actually doing? Because their activity is ultimately what impacts how diet is digested, but it also impacts potentially how the host is affected and the health outcomes in the host are affected. So we are using a technology that is called metaproteomics. And, I have a slide for that. It's would be slide 6. If I want to walk you through that real quick.
Please.
So as a starter, right? So the technology relies on measuring as many or all proteins in microbial communities. Proteins, of course, being what gives activity to cells, right? So if we know a protein is very abundant, it tells us that that activity is likely a very dominant activity of that particular cell. Whereas if there's, sometimes we know from a genome there's a gene there, but if it's not actually expressed into a protein, it actually is not doing anything, right? So we really learn about what is actually be used of the tool set that we have in a genome. So what we do is actually an integrated metagenomics, metaproteomics approach. So the first half of the approach is really the metagenomic part, which is essentially we're taking samples, we're sequencing all the DNA in that, and then we're trying to reconstruct microbial genomes from that. And there's a long workflow that we don't have to go through. But ultimately, we end up with hundreds of partial or complete microbial genomes by sequencing this DNA from a microbiome. And then from these genomes, we can predict which proteins might be there. So we can say this is the potential the microbe has. It could be doing X, Y, and Z. But we don't actually know if they're doing it. So then the other part is we take the same samples and extract all the proteins from these samples. And then, we digest the proteins, we turn them into little peptide snippets, and then we use a high-resolution mass spectrometer to actually measure all these peptides that we get from the proteins. And, we measure them in multiple ways. We fragment them to get also like information about the actual sequence. And so what we can then, and every time we measure a sample, we generate, say, 100,000 high-resolution mass spectra for each sample. And so then we take these spectra and compare them using computational tools to the protein sequences we predicted from the genomes. And then we can say, oh, this spectrum actually matches this protein. And then we do this, for hundreds of thousands of spectra, and then we can actually quantify. And then we can say this protein is actually there in high abundance, low abundance, medium abundance. And then, of course, if we have multiple samples, we can start comparing between samples. How does the protein or the organism that the protein came from change between samples? So that's kind of our powerful approach. So usually we end up with quantifying 5,000 to 10,000 proteins per sample.
For listeners and viewers, we now know when we talk about genomics, we talk about proteomics, and we talk about transcriptomics. What's interesting here, listeners and viewers, is that we're actually looking at the proteins. Many, and those include many enzymes, for example, things that might break down dietary components. But what's interesting here is that many folks have often looked at RNA as a sign of gene expression, but that's just part of the central dogma. As you just heard, going to the actual proteins lets you know what's being expressed. Yes, there are some RNA molecules that are important, but the enzymes, proteins that carry out most of life are obviously where you want to put a lot of emphasis. So that was a wonderful explanation. Thank you.
Yeah. Now did you mention RNA? It's like a little, yes, it's currently a problem in the literature that maybe we should mention briefly.
Please.
Is that people do transcriptomics, which is a great approach, but then they speak about gene expression, which is problematic in that, of course, as you just said, right, measuring transcription is just measuring one of the initial steps in gene expression. When we actually want to look at actual gene expression, we have to look at the final products, which can be the proteins, or it could be functional RNA, such as an rRNA or tRNA. But that is very muddled in the literature. And often when I'm a reviewer or an editor on articles, I see that some reviewers then get upset. You actually did not measure gene expression, you measured transcription. But it seems like that knowledge is almost lost sometimes, that doing transcriptomics does not actually mean that you have really measured gene expression, because you don't actually know if it's really expressed or not.
I really want to thank you for saying this, because as I mentioned, I'm going to be teaching in the fall, and sometimes we get carried away with, for example, doing promoter fusions to look at gene activity. But that's not the whole story, is it? And it's because sometimes it's hard to measure the activity of a protein the way we used to be able to do with, say, the lactose degrading enzymes that you find in the lac operon. The problem there is that you can measure, for example, beta-galactosidase activity. That's not difficult. But if you're looking at the constellation of different types of proteins with different functions, there's no way to do that. And that's one of the powers of your approach, in my opinion.
Yes. Another reason, thank you for saying that, yes. Another reason why I think our approach is particularly powerful when we now specifically look into the gut environment that I like to emphasize is it allows us to not only measure the microbiota and what functions they are expressing and what are they doing, we actually also measure any proteins that are derived from the diet. So we can say something about what happens to dietary proteins throughout the passage through the gut. But then also, it may come as a surprise to some, but not all, we also measure hundreds to thousands of proteins from the host. So there's a lot of like digestive enzymes that are injected into the gut, but there's also a lot of cell shedding. And we are seeing now over time that actually inflammation and other causes can actually lead to like very clear signals of what is going on with the host health or the host gut health based on host proteins that we are detecting. in these samples. So with just one method, we're detecting 3 different major players in the gut environment. Whereas, for example, if, coming back to transcriptomics, if you did transcriptomics and measured just transcription, you would only know about what the microbes are doing because you would be harvesting their transcripts. But of course, RNA from the host or RNA from the diet would be likely long gone by the time you would measure that in a stool sample. But we see all of these 3 players in our samples using that approach.
Would this be relevant to, and I may be using this term incorrectly, folks who talk about, quote, leaky gut syndrome?
I'm not, not an expert on specifically leaky gut, but we have looked, for example, at samples from inflammatory bowel disease, or where some inflammation is artificially introduced in mice. So we're looking at human samples and mouse samples in that regard, and yes, we do see massive increases in inflammation markers, massive increases in certain immune proteins. We see proteins sometimes related to blood, if there's real bad inflammation with lacerations, you can actually start seeing those proteins. So there's very clear markers for these things. And so for leaky gut, I would imagine that we would also see specific types of proteins that are indicative of that occurring.
So this is something that I remember many years ago, people were talking about looking at various proteins to be found in the blood that are leaking from cells, or leaking from the body, I should say. And ditto now. And this is, again, the power of this approach, because not just the microbes, but sometimes the host effects too. So I'm gobsmacked. It sounds great.
Yeah. Although sometimes people make fun of me for overselling it, and it can't do everything, but I do think it can do a lot.
Yeah. Well, I mean, everyone does that. No, this is wonderful. Could you walk us through a really representative experiment?
Yes. So maybe the one study I would maybe like to talk about is one that we have been doing for several years and is finally coming to its culmination. So if we look briefly at slide 4, that is kind of giving the major hypothesis, right? So based on the literature and based on our own observations, we came up with this hypothesis that the dietary proteins from different sources differentially interact with the gut microbiota, and that might ultimately, impact the composition and function of the gut microbiota, but then also impacts the host. And so what kind of drove us towards this hypothesis was that there's a lot of evidence from very large scale population health studies where they measure, follow people, hundreds of thousands of people over very long periods of time with surveys and measuring health outcomes. It's been shown that the source of protein that people consume does have a very large impact on their long-term health outcomes. Mm-hmm. So, for example, one of the findings that baffled us, and now we actually see some evidence on our side, is that evidently egg white protein, or egg protein in general, that is thought to be a really beneficial protein in these large-scale population health studies, actually has pretty bad outcomes. It's actually the worst protein in terms of longevity outcomes. So how they always express this is if you were to substitute plant protein for specifically egg protein, you would see a reduction in overall mortality risk by 24%, which is a massive number. Mostly when people think about impacts of protein, right, they think about red meat, which is known to have potentially detrimental impacts. The number for that is actually 13%. So if you were to substitute plant protein for red meat protein, then you would actually reduce overall mortality risk by 13%. So the impact of egg is actually much larger, which surprised us. But once we saw our own data, and we'll come to that later, we were like, wow, that is impressive. So based on those prior studies, we were very interested in looking at what are the impacts of specific sources of dietary protein on the microbiota, because we speculate that some of these interactions actually drive some of these long-term health outcomes. And so we designed an experiment, and I have a slide for that as well, which is slide 5. where you can actually also see the main people that did the actual work, right? I'm like conceptualizing, but Ali, Alfredo, and Aisha are actually the people that did the actual work. And so what we did, we developed an experiment where we made fully defined diets. And that's the nice thing you can do with mice, right? So we said, there's only one source of fiber, only one source of sugar, only one source of fat. And then we varied the source of protein. And we used ultra-purified protein. So it's really just a protein and not whatever else would come with the source of protein if you ate it naturally. And so we had multiple groups of mice, as you can see here. And then we were feeding these mice the different defined diets, each for like 7 days over time. And then always collected a stool sample at the end of each diet, so we could measure that later. And then we also had the same diets that we had in the beginning at the very end of the experiment to control for the fact that we want to see that the effects we're observing are actually really due to the changes in the protein and not just because time has gone on. I can say that fortunately that did turn out to be true, right? We went kind of back to the same level at the end of the experiment. It's definitely, we learned a lot also in terms of experimental design. We would do this differently now, but this is what we designed at the time. Nowadays, we would actually use more of a crossover design to improve this further. So we learned a lot of valuable lessons while doing that. And so we did all of this both with conventional mice that had a normal microbiota and with mice that were germ-free, so we could actually differentiate effects on the host that are based on the microbiota from effects that are just effects of the source of protein directly on the mouse host. Yes, so that's the experimental setup. And then we can talk a little bit maybe about what came of that. I already talked about the workflow. So what we then did with these stool samples, right? We employed this metaproteomics workflow integrated with metagenomics. So we sequenced everything. We ran everything on a mass spectrometer. Spent many years analyzing, that's the thing that is often, developing the experiment, measuring the samples, took time, maybe say 6 months. But what really took the time was analyzing the data. That took years and years. And so, so as I said, we couldn't look at all the different components. I'm going to focus on the microbiota today, but maybe just one glimpse briefly into what happened on the side of the dietary proteins, or the proteins in the gut in general in total. So I have a slide here, this is slide 7, where you can actually see the sum total abundances of different protein that we measured, right, out of the total protein that we measured in these samples. And so you can see we measured host protein, dietary protein, and microbial protein. And in most cases, actually host protein is very dominant in stool samples, right? In most cases, that's the most dominant. But there were some surprises here. For example, if you look at the rice protein diet, actually we measured huge quantities of rice protein, and that's more dominant than the host protein in this particular case. Or if you look at the yeast protein diet, you actually see that we have much more microbial protein than anything else that we measured in these samples. So actually the ratios shift quite a bit. I'm not going to, I have a little QR code here if this interests you more, because of course we started looking at each individual protein within these proteins, especially for the dietary proteins, and we see actually massive differences in how proteins get digested. That's actually one thing, maybe to realize is dietary proteins in reality, like say we have a soy protein isolate, they are actually made out of 1,000 different proteins. And when they go through the gut, some of them get digested very easily and completely disappear, whereas others get enriched because they're evidently not digestible. Some of these proteins actually have potential physiological impacts. Some of them, like a trypsin inhibitor, might mess with your digestion. There's some that are antimicrobial. They might mess with your microbiota. Things like that, right? So there's a whole world just to explore in the realm of the fate of the dietary proteins themselves. But so enough of the proteins. So we can look a little bit into what happens to the microbiota. It's a very large story, so I'm going to show just a few highlights today. So for example, if we look at slide 9, we did different measures of like what happens to the microbiota in terms of overall composition. So really looking at who is there. And so there's on the left side, you have a graph that shows you alpha diversity over time, over the different diets. Alpha diversity really just being another term for saying how many different microbes are there, right? And the higher the score, the more different microbes. And you can see there's actually some massive differences, right? So for example, for the yeast protein diet, alpha diversity really drops, which tells you diversity disappears. There's only very few species suddenly. And similarly for the egg protein diets, we have a massive loss in alpha diversity. Another thing we can, of course, look at is the ratio of microbial proteins to host proteins. And here we see that actually that changes. And, I had mentioned this before. For the yeast protein diets, the ratio of microbial proteins to host proteins goes up a lot. So they really accumulate a lot of mass. And then there's a measure that it's a little more complex, which is beta diversity. It's essentially a measure where we compare the types of microbes that are present in two different samples. And so what we're here doing is we have this casein soy diet at the beginning. And so essentially we have multiple of those samples, and then we're comparing all the samples to all the casein or soy diet samples. So say when we compare then in the blue line, we have the soy protein microbial composition, and now we compare the yeast protein microbial composition to that, and we see there's a massive difference, right? When the value goes up a lot, it means it's very different. So the microbial community composition in the yeast protein diet is very different from when we have soy protein or casein. And here again, the egg protein diet also has a massive shift in a microbial community composition. Now, the nice thing about this data is, right, we can look at this from a very high level, overall changes. But, if we look at slide 10, for example, we can drill a lot into the depth of this, right? We can say, okay, let's look at specific microbes, because by doing this integrated metagenomics, metaproteomics approach, we have the genomes, or which are units of microbial species, essentially. And we can relate all the proteins to that. And so we can quantify individual microbial species in all these different samples, and you get this very nice heat map. I really like that one, even though it looks very complicated. But essentially what we're showing here is the microbial species that are the most abundant in any of the samples, right? So that's 30 most abundant ones. On the bottom, you see all the different names of the samples, and we have a little color coding for the different protein sources. And I drew a little box around it for just the samples, and the samples here are in the columns, right? Each column is a sample, and then across we have the species. And, the color indicates the hotter it is, the more of that species do we have. And so here I highlighted in that box the yeast and the egg white protein diet, and you can see they all sit together, right? So the algorithm decides they're all more similar. That's why they all cluster together, and you see this little tree on the top that actually has them all on the same kind of branch. And, there's one organism that is particularly hot. That's the Bacteroides theta iota Omicron, right? So that organism really seems to be driving some, is what's driving the difference of these particular microbial communities under that protein diet. And that's one of the ones that we looked at in more detail. But, we can go deeper and deeper in this data if we want to. So now that's, of course, all the stuff we could also have determined by just sequencing 16S genes, right? We might have gotten to this place. All of this, however, is done with metaproteomics, right? We're actually not looking at 16S sequences. We quantify these microbes based on the abundance of the proteins. So we're essentially measuring their mass, not the number of their genome copies. So a bigger microbe would be counted much more than a smaller microbe because it contributes more mass. Even if they have the same cell number, it would look more dominant. And so what we can do next is we can drill into function. Now we can actually ask, what are these proteins doing? And so, in slide 11, there's an overall view of all the proteins independent on which species they're in. We classify them by big picture categories, say the amino acid metabolism or gene expression. And then we just summed up all the abundances of all the microbial proteins just to see what's the overall function. And when you look at this, I mean, just to highlight a few things where we actually see some big differences is like, for example, if you look at glycan degradation, there's two sources of protein where this particular functional group changes a lot. That's under egg white protein diet and the yeast diet, right? Much more glycan degrading enzymes are there, much more abundant. Whereas, for example, in gene expression, you would see that in the egg white protein diet, gene expression overall, any genes that are involved or proteins that are involved with it are dropping quite a lot in terms of abundance. So if you look at this, there's a lot to explore in terms of different things. And the nice thing again now is we can drill into more detail, right? Because we know each of these proteins. So say if we go, of course, one of the basic questions when we deal with protein is like, what happens to all the amino acids, right? Because when we think about it, initially it's just strings of amino acids, which we later realize is not quite true. But so what happens to amino acid degradation? And so if we now look at slide 12, we can say we're only looking at all the proteins that are involved with amino acid metabolism. And if we want to be more specific, we can look only at the proteins that are involved with amino acid degradation. And so when you look at these graphs here, like these, you see these little dots. Each dot is a sample. And then you see the little boxes around, which are like confidence intervals. And so essentially, when boxes don't overlap in a graph, that means they're actually really different, significantly different. And you can see that, for example, in the rice protein diet, when we look at amino acid degradation, rice protein diet, the degradation proteins are much more abundant than in any of the other samples, right? Similarly, we see some increase there for the egg protein diet. Now, that is curious, right? As I mentioned earlier, the rice protein actually seems to be not well digested, so clearly makes it to the colon where most of the microbiota sits and ends up in the stool. And surprisingly or interestingly, we also see a much higher amount of amino acid degradation. Now we could speculate that this is due to the fact that the microbes are actually seeing more protein that they can deal with. Because generally the idea is if a protein is very well digestible, it gets all absorbed in the upper intestinal tract, and the microbes never have much contact with it. Good. I mean, I don't want to, I can go to another slide, but I'm not sure if you have any questions in the meantime. I don't want to monologue.
Well, no, I just want to, summarize the power of this to listeners and viewers. I do, and I think you did a wonderful job explaining things, but I had a question. Just as you can get fooled by transcriptomics by just looking at RNA, is it possible that degraded proteins that are non-functional could confuse you with your approach?
That is actually a very good question. Yes, it could be. So that question came up for us, especially like when we look at the fate of specific dietary proteins.
Sure.
We say some of them have functions that could impact the gut, say like antimicrobial activity or inhibiting digestive enzymes. But of course, we cannot say based on the we can say these are still abundant when they come out in the stool sample. So clearly they're there. But we cannot say, are they still correctly folded? Are they maybe like broken up? Are they functional? We cannot, based on this method alone, we cannot say that, right? That is correct. So we would have to go and do, say, follow-up work to then see if these proteins are actually intact. Say, like if it's lysozyme, which is one of the proteins we're curious about, which degrades microbes. We could take a stool sample, do some experimentation. We could say, oh, okay, lysozyme is still active when we like spread it on a plate and see that it digests microbes. Yes, but it is a limitation.
Oh, absolutely. And I do not mean to be critical. It's just something that occurred to me with it. Thinking about the research that your group has done, Manuel, do you have any expectation that the results might translate into recommendations regarding diet, say, and fiber supplements or types of protein?
That's a good question. I wouldn't want to overstate our results, right? I think we have to see this in the bigger picture of things. I do think our results contribute to an emerging picture of what dietary protein sources may be healthier than others and under what circumstances. we are taking this work now, for example, into the direction of research on inflammatory bowel diseases. And I do think ultimately, in the context of all the other studies that are happening, our results will definitely help inform dietary recommendations for specific health purposes or for specific diseases.
Now, that would have to be very gratifying to be part of that. I'm not a gym goer per se, but I sometimes go, and I'm trying to be on a treadmill like 6 or 7 times a week. And there's this whole issue called bro science, which I hadn't heard of until just today, in fact. And some of the folks that are really interested in weight training have particular types of protein supplements they're interested in, dietary considerations. Do you think that what you have found could be translated into recommendation for those individuals?
It's a very pertinent question. I have a teenage son who is very much into going to the gym. So we have a lot of discussions about protein. I can see this going in multiple directions. One thing that I've learned through discussions with my son is that there's a lot of misinformation in terms of protein, right? People seem to think that there was this like very ancient, never proven idea that nutritionists still teach today that specific types of proteins are better for building muscle or anything like that. And that is true if you're like under kind of a starvation situation. If you have very limited access to protein, it really might matter the exact amino acid composition of the protein matching your body so that you can at least make the most of it. But generally, in our society, we are not protein limited, generally. So at that point, if you have just enough of everything, it doesn't really matter what the exact composition of protein is. There's also some more outrageous misconceptions in my perspective where like some people claim that plants don't even have all the amino acids. And I'm like, where do you think they came from in the animal? Of course, plants produce all the amino acids. So that's, I think, one of the perspectives. Maybe people listening to that think more carefully about some of the myths surrounding protein because there's a lot of misinformation. The other perspective is, and that's where I think people should maybe be a little alarmed, because depending on what your preference is, some people take whey protein, some take casein, some take eggs, some take just raw meat, red meat. And our research and the research of many others shows that there's definitely concerns with certain types of animal proteins, right? Casein, for example, I think might be fine. It's a protein that is made by the mother cow to be actually consumed by a mammal. But other proteins have shown detrimental side effects. So, even while you're young, you might not have those effects. If you do think long term, what about, the link between red meat and colon cancer? Is that something you should maybe consider? So our research kind of reinforces this. We see these massive impacts of these proteins. So that's something to consider. Of course, other questions, is it nutritionally relevant? We are showing that plant proteins, or at least some of them, right? It's not really necessarily just dichotomy because each protein seems to have its own effect. But plant proteins or specific types of proteins seem to have more beneficial effects on your microbiota and may ultimately have better long-term outcomes. And nutritionally, they're often equivalent.
So I'm really interested in this and the study about egg protein leading to a negative effect that you mentioned earlier in terms of lifespan. Are you yourself now vegan?
I've been vegan for quite a while now. It's not directly related to these studies.
Well,
but we've come as a family. Over time, we've gone through vegetarianism, and I am now vegan, yes.
So it is interesting when people talk about diet, and I think a lot of listeners and viewers will be interested in this.
Our results, of course, have to be seen in the bigger picture of other results, right? Because we are mostly finding, at the moment, crazy effects on the microbiota. Something is happening in terms of changing function and taxonomy. We of course, we can move on, briefly talk about what's happening on the host side because we looked at that as well. And we actually see massive, which I did not anticipate, but there's massive changes in what's happening in the host proteome that indicates that actually some of these protein sources may have simulate antimicrobial attack or stimulate inflammation in the end. Maybe before we come to this, I could maybe talk about one of the most surprising findings in regards to the egg protein.
Please.
Because we were like, what's going on, right? This cannot just because it's a string of amino acids. And so I mentioned before when we look at slide 13, I mentioned before that one of the things we found is that glycan-degrading enzymes are particularly abundant in the yeast and the egg protein diet. And so glycan-degrading enzymes are essentially enzymes that take, chains of sugar, for example, starch or cellulose or other things, right? They are the ones that cleave so-called polysaccharides or glycans, right? Long strings of sugar, which we usually think of as fiber and starches and things like that. And so these are specialized enzymes that can cleave different, if you have different sugars that are bonded to each other, you need a different enzyme for every single bond. So we were like, wow, why is the source of protein have anything to do with sugar chains, right? And so we looked at that in more detail. So in the bottom right of that slide, you can see just the abundance of all the glycan-degrading enzymes. And we can see that for Bacteroides theta omicron here in red, we see that that's actually the main driver of that change. That's the one that has all these glycan-degrading enzymes. If you look at this interesting square bubble plot, you can see actually all the different glycan-degrading enzymes and their abundances across the different diets. And you can see each diet has its own little pattern in terms of which of these particular proteins are abundant. So clearly something is going on with B theta using these enzymes in response to dietary protein. We were like, what is going on? And so we dug into that into much more detail, and I'm not going to go all the way right now, but maybe if we look briefly at slide 14, there's on the right side, you see a so-called volcano plot that shows essentially all the different proteins we detected. And we see the fold chains just between the two diets, the yeast protein diet and the egg white protein diet. And you can see there's massive differences. So B theta is clearly responding with different glycan-degrading enzymes to different sources of protein. And to make a long story short, essentially, in the aftermath, of course, you want to slap your forehead. A lot of these dietary proteins actually have a lot of so-called glycosylations. So there's long strings of sugars that are attached to these proteins. And of course, what microbes are doing, they're like gnawing those strings of sugars off, right? So they cleaving them off and then they use them as a substrate to grow. And since each of these sugar side chains is very different in terms of its composition and structure, they need different enzymes depending on what kind of sugar side chains they are digesting. Now, so that was like, wow, okay, so one of the effects of dietary protein source is actually driven not by the amino acid backbone, but what's hanging on the sides. And what was very particularly interesting for us for the egg, and this is something we have to follow up, so we shouldn't, is that the same enzymes that are known from this organism to actually be used to cleave off sugar side chains of a protein in the gut, which is called mucin. That's a protein that's basically putting slime on your layers of the intestinal tract to kind of help things move along and prevent things from getting into the actual tissues. And so these enzymes are actually used by this bacterium to actually cleave proteins and sugar side chains of these proteins in this mucin, which is kind of, fine. But if they do too much of that, then they might actually start depleting that layer. And so we found it quite interesting that the same exact enzymes are used for egg white protein. And so egg white protein actually has a protein that's called ovomucin that is structurally, at least for these sugar side chains, very similar to the sugar side chains of the mucin in the gut. So, there's correlation, not causation yet, but there's we're starting to see a picture that might make sense, at least in one direction, why egg white might, for example, stimulate organisms that then also start degrading the mucus layer. There's, of course, other aspects to egg white protein, such as the lysozyme that I mentioned earlier, which is actually a major killer protein that just generally destroys microbes in your microbiota, which might also have some part of that effect.
This is so fantastic to hear about, because as I was listening to you throughout this talk, the idea that you're leading to a particular bloom of microbes in response to dietary changes makes sense to me, because yes, we're a tube and there are ecological niches along that tube, and the GI tract is one of them. But one of the things that always fascinated me is the idea of B theta as, please forgive me this, a mucus farmer. Because you're right, it's able to cleave off stuff from the mucin, and if it gets too good at it, that's bad, yes. But there's some evidence, I believe, I don't have a reference for you, that it tends to stimulate mucin production. And that's what I thought of as you were talking about this. But it turns out that those signals are there from the diet too, which is fascinating to me.
Yes, that's I like the mucus farmer. That's a good term. I think, yes, and I might be wrong, but, yes, B theta is doing that. There's another organism called Akkermansia mucinipila that is also kind of farming mucin that we have some reason to believe is actually also gets stimulated by egg white protein. So there's a lot more depth to what we can discover there in terms of how the dietary protein actually ultimately changes the microbiota and actually influences potential host processes.
Fascinating. Oh, this is really exciting to me. So I loved hearing about a surprising result that you drilled down in. Okay, is there a particular investigation that you want to embark on based on this in the near future? In other words, what you're doing now.
Yeah, that's a good question. So, yes, we're actually following multiple different prongs there. One thing I mentioned is, we're looking at the host as well. And so we have some nice preliminary results where we see really massive differences in, say, antimicrobial proteins from the host, immune proteins from the host, where we see really big changes in germ-free mice. So not even in linkage with the microbiota. Well, these proteins change. And then once you add the microbiota into it, then it gets even more. It's hundreds of proteins are changing extremely strongly in response to the source of dietary protein. So that's one avenue we are pursuing, where we actually now have an upcoming collaboration with people at UNC and Stony Brook that actually work with human studies, where they actually have human volunteers that are actually eating a diet strongly enriched in pea protein or egg white protein, where we're hoping to look at some of these host responses as well, to see if we see the same thing happening in humans, because then that would make it really even more relevant. So that's one of the things. The other thing is we're really excited about these egg white protein results. Of course, there's a lot where we speculate on causality and mechanism, but we really want to now do experiments, for example, where we take just lysozyme or avidin, which is an antimicrobial protein from egg white, or eggs that are prepared in different ways, and either feed, these things to mice, when it has to be fully controlled, or ultimately, we're very interested in, does the way you prepare your eggs ultimately matter for how that impacts you, which would, of course, be nicest to do with human volunteers. Because, for example, the lysozyme, which is this enzyme that just kills a lot of microbes randomly, is heat stable. But how heat stable is it? Is a soft-boiled egg your problem, or is fried eggs also your problem? Things like that, where it becomes really relevant at what point do some of these effects appear and disappear. So that's another study. And then a third kind of a study that we're following is, one limitation with these very defined diets is that, for example, there's not much complexity in fiber. And we know from other studies, right, fiber is one of the main driver of diversity of gut microbes. Mm-hmm. And so we want to start now doing combinations of different fibers with different sources of proteins to really better understand in a more realistic diet setting do we see the same effects? Do the effects change? Can they be ameliorated? Are they getting worse? Right. So, adding a more beneficial fiber like inulin, would that help? And then one of my students is also very interested in the question, for example, if you now combine, say, beef with egg white, is it getting even, is it just, summative, additive bad effects, or is it actually together the sum of the total is even worse, So there's a lot of interesting questions we can pursue. And then hopefully, ultimately, that will lead to some recommendations that can be made.
It's interesting because many people view microbiology as a field, or microbial sciences, excuse me, as a field where we focus mostly on pathogens, or my end was environmental, microbes that do things in nature that we're interested in. But it turns out that there's this overlap to human health that doesn't really have to do with even though Arturo Casadevall does not care for the term pathogen, you know what I mean, organisms that can cause disease. Now we have a situation that is really interesting because there could well be not one cause for things, but groups of different effects coming together. And because we're kind of reductionist as scientists, I find this interesting to think about the sum total of things working together. And now we have the technology to start picking it apart and learning more, as you have just described.
Yes, that's definitely one of the things we do in my lab, is trying to cover this whole spectrum of, very reductionist, but then looking at systems, these not abiotic systems, where we can start, okay, what if we do 2 microbes? Mm-hmm. And then, of course, going to full life field settings or into humans or mice. And we're seeing, we're doing a lot of work, which we didn't talk about at all in plant microbe interactions in the root. And we're seeing this a lot, right? If you have a single microbe inoculations, you already see really interesting effects on what the plant is doing, what the microbe is doing. But now you add like 1 or 2 or 3 microbes together, you see that they're completely changing what they're doing depending on who else is there. They might be helping each other. They might be competing. They might be, having an effect on how well others can colonize, or how abundant they're going to be, how long they're going to be persisting. So these systems really allow us to study the interactions, and we do have to take it apart. I think it's just sometimes too complex when you look at everything.
Well, and I mean, the technology improves and changes. I remember when biofilms were first being studied, and I was reading some of the work and reviewed some papers, and I would comment, you're not going to find biofilms in nature composed of one organism very much. And their response was, well, now we're working on 2. But you see, that was what we could do at the time. And now you look at people who are doing, like Yvonne Cooper has some wonderful work where he's looking at some of the complex biofilm communities in the human lung. So as technology improves, we get less and less reductionist and more ecological, maybe is a good word.
Yes. That's where we had it. There's still a lot to explore.
I want to emphasize to listeners and viewers that there is such breadth in the Kleiner Lab group, and notice again all the credit that's being given to all the coworkers. That's something I really want people to understand, is that microbiology is inclusive and it's a group effort, and this is a really great example. Getting back to my point, in the show notes will be a link to the laboratory website, which has so much different stuff, including things that maybe I can have you on again to talk about plastic degradation. I know a lot of people are interested in that. But my point is, there's such a breadth of things there to look at and think about. But you just mentioned this, sometimes there are commonalities. You mentioned what you find around the roots, the rhizosphere, to what you might find in the gut. And maybe there are themes in common, evolution playing different tunes but the same melody, for lack of a better term. I have that backwards, I think. But anyway, you get my point. And that's something that's really a wonderful thing to follow up on in the future. Do you think that you could spend a couple of minutes talking about your path to the microbial sciences?
My path to the microbial sciences, yes. Yes, and thank you for pointing out my team. If you want to show the slide I had prepared, you can see a picture of my awesome team at one of our group retreats at the North Carolina coast, where we like discuss a lot of science, a lot of fun. That I do recommend group retreats once in a while. They really invigorate the team and generate synergies. And also you can see our beautiful logo that one of my PhD students made that I'm very proud of. So my path to microbiology, I'm not going to tell you my whole life history, but I did a lot of things before I became a microbiologist. I grew up in Germany. I did my degree there. I did civil service, which was at the time mandatory, military service or civil service. So I spent some time that I did actually study design. So I studied design for like a year and then realized, it was really just to try, can I be a photographer? Because I really liked that, but I always was really good in natural sciences. Then I decided I could never be a freelance photographer. That's a tough job. And then I, traveled the world for a year. I sailed across the Atlantic Ocean and things like that, which was a lot of fun. And afterwards, I was kind of inspired to think about, I want to really work on something that is important and helps with the environment. So I was looking in actually studying something related to environmental sciences. What I ended up with studying was actually biology. So at the time, we had like 5-year degrees in Germany. So there was a 5-year diploma in biology. And I was like really headed towards some environmental thing there. But what was nice about the degree is you really had to learn everything, right? You did like 2 courses on plant physiology, 2 courses on animal physiology, botany, plant determination, everything, zoology. And I was actually, at the time, I knew nothing about microbes. In school, we didn't really talk much about them. We had a little bit of the genetics and stuff, but it really was never a thing. And then we had a course where we got like kind of the systematic introduction to microbiology. And of course, systematics, maybe not as exciting to me, but what was the kind of eye-opening at the time was what these microbes can do. All these extremophiles, all these different things they can eat, all these different things they can make. I was like, wow, that's it. And so then I started to go more towards the microbiology track. And I was at the time at the University of Greifswald, which is big and famous. They had a big group for proteomics, very renowned across Germany. They were working on Staphylococcus aureus and Bacillus subtilis, which, interesting, very important model systems. But I was really like fascinated by microbial communities. And so in my studies, I had made a lot of headway that I had some extra time on my hands because I was had extra time on my hands, essentially. So I went and did some internships. So I went on an internship in the US with Greg Walliser working on Myxococcus.
Right.
I went on an internship to Regensburg at the time. So I met Nicole de Villiers, who is working on these very fascinating organisms, and I was able to strike a deal with her and my advisors in Greifswald. I said, I'm going to stay in Greifswald. They really wanted me to stay. It was very flattering. And they were willing to let me work on a system totally outside of what they were doing. And so I studied the symbiosis in marine worms for my diploma, and really got involved into this realm of microbial symbiosis, microbiomes. Then I went on and did my PhD with Nicole de Villiers. And from there it took off.
What's interesting here is, and this is a common theme with guests of the podcast, it's not something that's terribly straightforward often. And I always tell students, I shouldn't say warn, I always tell students to be prepared to take opportunities as they occur. There's that Pasteur joke that chance favors the prepared mind. Just take the opportunity. And give it a try. Don't talk yourself out of things. And you're a really great example of that. And you had these different opportunities and directions, and you took, and look where you ended up.
Yeah, and I love that quote. That's a Pasteur quote, right? It's very big at Indiana University on a building. I love that quote. Yes, and there's a lot of serendipity. So, for example, this meeting with Nicole de Bellier was not actually doing an internship with her, but I lived in the institute for my internship. And at night, at 11 at night, I was like walking around the institute, going to the tea kitchen, and there she was working late. And so we struck up a conversation. And from that, basically developed my whole career later on, just from that chance meeting that I ran into the right person. And, we were prepared for that meeting, I guess.
There are no coincidences, only opportunities.
Yeah.
Well, I want to really thank you a lot for this opportunity. Once again, I'm impressed by the depth and breadth of your studies in the microbial sciences. It's hard on me because everything interests me. And I always want to know more, but I'm glad that we stuck to this particular topic because I think it's things that will it's about things that will interest many, many people. And I am very grateful for your time today. And I want to take this opportunity to wish you and your colleagues and family the very best.
Thank you so much. Thank you so much for inviting me.
It's really been an honor. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, epic links as always, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Manuel Kleiner is Associate Professor of Microbiomes and Complex Communities in the Department of Plant and Microbial Biology at North Carolina State University in Raleigh, North Carolina. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our quality quorum for today. See you next time on Matters Microbial.